There's a weight to stillness that most people don't notice until they stop moving long enough to feel it. Not exhaustion exactly, something older than that. Something the body seems to already know even when the mind doesn't catch up for a while. You found your way here again, Sleepy Biologist. And if this is your first time, something brought you to a channel about biology and sleep at this hour, and I find that quietly interesting. The people who end up here tend to be the kind who can't fully let a question go. That's worth something. This space exists because enough of you felt that way that it became real. Channel members, supporters, those who've left comments in the dark hours. Thank you for keeping this particular quiet alive. If you've been here before, you already know how this goes. No rush, nothing to retain. If a thought drifts past and you lose it, that's fine. That might be the point. Tonight we're going somewhere most people never think to look. Not deep ocean, not forest canopy, something less photogenic than either of those, mud. Specifically, the layer of sediment beneath still water. The anoxic zone, if you want the term, though the term isn't what matters right now. What matters is the texture of it, the lightlessness, the particular kind of chemical patience that accumulates in places where oxygen runs out and something else takes over. Think about what happens to a lake in autumn. The surface temperature drops. Water layers that stayed stratified through summer begin to mix slowly, and what falls from the upper water, organic matter, the remains of algae, bits of leaf and debris, sinks. It settles. It lands in sediment that hasn't seen light in years, and there it sits, under pressure in cold that doesn't fluctuate the way surface cold does. You breathe because your mitochondria need a final electron acceptor. Oxygen is that acceptor for you. It waits at the end of the electron transport chain and accepts the electrons your cells have been passing down a gradient. And the energy released in that process is what keeps you warm, keeps your heart moving, keeps the pressure behind your eyes stable enough that you can close them now. In the sediment below still water, oxygen runs out within the first few millimeters of depth. Below that threshold, everything that wants to respire has to find another acceptor. And iron, oxidized iron, ferric iron, the rust-colored iron bound into sediment minerals, turns out to be available. It's abundant in many sediment types. And for certain organisms it works. The electron still moves. The gradient still releases energy. The cell still lives. The acceptor is just different. There's something about that substitution that takes a moment to settle if you let it. The machinery of respiration, the basic logic of it, is the same. Move electrons, capture energy, continue. What changes is only the molecule waiting at the end of the chain. Iron instead of oxygen. The sediment layer where this happens isn't dramatic in any visual sense. It's grey, or brown-grey, finely grained and dense. If you pressed your fingers into it, and you wouldn't want to particularly, it carries the smell of hydrogen sulfide. Faintly the egg smell that anaerobic chemistry tends to produce. You'd feel resistance, then slow give. The kind of surface that doesn't spring back. Below that surface, chemistry running without interruption. Electron transfers occurring in the dark at rates that don't respond to whether it's day or night above. The organisms doing this are too small to see, too numerous to meaningfully count in any single sample, and completely indifferent, if that word even applies, to the fact that above them, an entire oxygen-rich world has organized itself around a different kind of respiration. I find that indifference interesting. Not as a statement about anything, just as a fact worth sitting with. The particular organism this narration is built around hasn't been introduced yet. That's deliberate. Before the name, before the discovery, before the laboratory and the electrode and the contaminated aquifer, the sediment, the conditions, the chemistry that was already running long before anyone had a reason to go looking, Geobacter. The name will come. There's no urgency in arriving at it. For now, what's worth knowing is simpler than a name. Somewhere in the sediment beneath almost every lake, every slow river, every waterlogged soil, electrons are moving. They are passing from organic carbon to iron. They are doing this right now, tonight in the dark, at a pace biology sets when nothing external demands it go faster. Your body is also completing electron transfers tonight, slower than when you're awake, quieter. The mitochondria in the cells of your diaphragm are still working, still moving charge across membranes, still doing the fundamental thing that respiration is, the same fundamental thing happening in the mud at the bottom of a lake. The acceptors are different. The depth is different. Everything else is closer than it looks. What the sediment holds beyond the chemistry is time. Not geologic time in the grand sense, just accumulation. Layer on layer of organic material, each layer representing a season, a year, a decade of things that fell and settled and were slowly broken apart. The organisms living in the deepest layers of that accumulation have, in some cases, been metabolizing the same carbon compounds for longer than the soil above them has existed in its current form. There's no way to feel that directly, but there's a kind of knowing that arrives when you stop trying to picture it and just let the fact rest. That beneath the surface of something that looks still, layers of time are stacked in chemistry, and something in those layers is still running. That's enough for now. The sediment doesn't ask to be understood all at once. It just sits and breathes in the way that things breathe when oxygen isn't an option. And underneath it, finer than anything visible, something is moving charge from one molecule to another, extending maybe a filament thinner than a human hair would be if it were divided 100 times over, reaching toward the next available surface, the next available acceptor. That reaching hasn't stopped. The word eat is doing something strange in this chapter's title, and it's worth admitting that up front. Organisms don't eat metals the way you'd eat something. No ingestion, no chewing, nothing that resembles consumption in any felt sense. But reduction, electron stripping, the act of pulling charge off an iron atom and incorporating that transaction into the basic arithmetic of staying alive. There's something in that process that it gets closer to than any more accurate verb does. So, what it means to eat a metal. Iron exists in two oxidation states that matter here. Ferric iron, Fe3+, is the oxidized form, the form bound into minerals, locked into sediment in compounds like iron oxyhydroxide. It carries a positive charge that makes it stay relatively inert, not going anywhere without chemical persuasion. Ferrous iron, Fe2+, is what you get after the persuasion. Geobacter is the persuasion. What the cell does at its most stripped down, it oxidizes organic carbon compounds, acetate, often a 2-carbon molecule that sits at the center of a lot of anaerobic metabolism, and it uses the electrons released in that oxidation to reduce ferric iron. The iron accepts the electrons. Its oxidation state drops from 3 to 2. The cell captures energy from the transfer, and the iron, now ferrous, dissolves. It moves. It enters the pore water of the sediment and becomes part of the chemistry of the layer, cycling back eventually into forms that other organisms or other reactions will touch. There's a smell that sometimes comes through old radiators in winter, metallic, faintly mineral, the specific warmth of the heated iron, and I'm not sure why that keeps surfacing as I think about this. It has nothing to do with what Geobacter does. The iron in sediment is cold and lightless and nothing like a radiator. But the mind goes where it goes. What's harder to hold on to, and this is the part that took me a few readings to feel properly, is the scale at which this is happening inside a single cell. A Geobacter cell is roughly 1 to 3 micrometers long. For reference, a human hair is about 70 micrometers in diameter, so you could line up somewhere between 20 and 70 Geobacter cells across the width of a single hair, and even then, the number wouldn't feel real in any tactile sense. Inside that cell, the electron transport chain is running. Proteins embedded in the inner membrane are passing electrons down a gradient from the organic carbon compound being oxidized through a series of carriers toward the outer surface of the cell where the terminal reductase enzymes sit. Those enzymes make direct contact with iron minerals outside the cell. The electrons arrive, the iron accepts them. This happens continuously, billions of times per cell per day. Not quickly, exactly. Biology at this scale doesn't feel quick from the outside, but without interruption. I think what makes this difficult to really absorb is that we tend to picture metabolism as something warm, something interior. The mitochondria in your cells are inside you, sealed away, protected, running their chemistry in a wet and private space. The idea of a cell conducting its respiratory process outward, across its own membrane and into a mineral sitting outside, that violates some instinct about where metabolism is supposed to happen. But Geobacter's electron transport chain ends outside the cell. The terminal acceptor, iron, is external. The cell has evolved proteins specifically for bridging that gap, carrying electrons from the inner membrane all the way to the outer surface and sometimes beyond it. The nanowires, the conductive pili, extend this reach even further. A later chapter will spend more time there. For now, just the basic fact. The chemistry crosses the boundary of the cell. It has to. The iron is out there, not in here. What this produces, as a consequence, is mineral transformation on a scale that sediment geochemistry has to account for. As Geobacter reduces ferric iron to ferrous, the mineral phases in sediment change. Iron oxyhydroxides dissolve. The texture, the porosity, the chemical availability of the sediment layer shifts, slowly, incrementally, across years and decades, but genuinely. The sediment around an active Geobacter population is chemically different from sediment where the organism isn't working, which means the composition of the mud beneath a lake is, in part, a biological product. The geology is being written by the microbiology. That's not a metaphor. It's a straightforward consequence of reduction happening at sufficient scale over sufficient time, and it tends to get left out of the way. People talk about sediment, which is mostly framed as a physical or chemical story. The biological authorship of it is quieter, harder to see. You've probably never thought about what's under the sediment of a lake you've swum in. Most people haven't. It doesn't come up. You feel the cold of the water and the way the bottom gets soft where the sand gives out, and then you turn around and swim back. But underneath that soft layer, and underneath the layer below it and the one below that, the iron has been moving, not dramatically, not visibly, just steadily, in the way that geology moves when biology is doing the work, patiently in the dark at a pace that has no particular relationship to anything you're doing on the surface. The cell doesn't know you were there. It was already running its chemistry before you arrived, and it kept running after you left, and the iron kept accepting electrons, and the sediment kept slowly, incrementally, becoming something slightly different than it was. There's something in the mundaneness of that which I find, I'm not sure comforting is the right word, settling maybe. The idea that beneath the places we move through without thinking, chemistry is running that has nothing to do with us, and everything to do with keeping the mineral and carbon cycles of the planet in some kind of working order. We didn't set it up, we can't maintain it. It was running before anything with a nervous system existed to notice it. And tonight, in whatever sediment lies closest to where you are right now, somewhere in waterlogged soil or the bottom of a drainage basin, or the anoxic layer beneath a pond you may have passed 100 times. Electrons are moving from carbon to iron. The cell is still doing the thing that EAT, for all its imprecision, almost manages to describe. The Potomac River. Somewhere between the slow water and the sediment beneath it, in a sample most people would have found unremarkable, a graduate student or a technician, probably, doing the careful and unglamorous work of collecting mud, something was present that hadn't been formally described yet. Derek Lovely was trying to understand ion cycling, not electricity, not bioremediation, not the future of microbial fuel cells or contaminated aquifer cleanup. He wanted to know how iron moved through anaerobic sediment, how ferric iron became ferrous iron in environments without oxygen, because that question, quiet as it sounds, had a gap in it that hadn't been filled. The gap was something biological was doing it. The rates of iron reduction in sediment were too fast, too consistent to be purely chemical. Something alive was accelerating the process. He found it. Geobacter metallireducens. The name came later, as names do, after the culturing, the characterization, the slow confirmation that what had been isolated was genuinely new. Geobacter from the Latin for earth. Metallireducens for what it does, reduces metals. The name is accurate in the way that an address is accurate. It tells you where something is, not what it's like to be there. What Lovely had found was an organism capable of coupling the oxidation of the organic compounds to the reduction of ferric iron, running a complete respiratory process using a metal as the terminal electron acceptor. In the absence of oxygen, in the dark, in mud. It had been doing this, presumably, for a very long time before anyone looked. There's a particular kind of scientific discovery that doesn't announce itself. No visible phenomenon, no dramatic instrument reading, no moment where the data produces something obviously impossible. Just a result that quietly doesn't fit the existing explanation, and a researcher careful enough or stubborn enough to follow that misfit rather than set it aside. I tend to think those are the discoveries that matter most in the long run. Not because quiet discoveries are inherently more significant than dramatic ones, but because they require a different kind of attention, a willingness to take an unremarkable sample from an unremarkable place, and treat it as if it might contain something worth knowing. Mud from the Potomac. What the discovery did to the field was gradual. The implications. That anaerobic iron reduction was biologically mediated. That organisms could respire using solid metal minerals. That the electron transport chain could end outside the cell took time to propagate. Other researchers had to replicate it, extend it, find Geobacter in other environments, discover related organisms doing related things in other sediments around the world. Science rarely restructures itself overnight. It adjusts slowly the way sediment does, one layer at a time, each new result pressing gently on the ones beneath it. By the mid-1990s, dissimilatory metal reduction, the formal name for what Geobacter does, had become a recognized field. By the 2000s, the discovery of electrically conductive pili had opened questions nobody had thought to ask in 1987. None of that was visible in the original sample. It was just mud from a river, collected carefully, handled correctly, examined by someone who noticed that something didn't add up. There's no tidy lesson in that exactly, not one that generalizes cleanly. Some unremarkable samples contain nothing that changes anything. Most do. What stays with me is the specificity of it. The Potomac River, 1987. A question about ion cycling, not electricity. The organism already present in the sediment, already running its chemistry, entirely indifferent to whether anyone ever cultured it or named it, or spent the next 4 decades trying to understand its conductive filaments. It wasn't waiting to be discovered. It was just there, doing the thing it does, in the mud, in the dark, at the pace biology moves when no external pressure is demanding anything of it. The discovery happened to the scientists. The Geobacter just kept going. Something extends. That's where this has to start, because the fact of extension, the reaching, is what the rest of this chapter is built on, and if you lose that image early, the biology becomes harder to feel. So, something extends from the outer surface of a cell that is already smaller than most people have a working intuition for, into the space beyond it. Not randomly, not passively. The cell builds this extension, grows it, deploys it into the surrounding environment as a functional structure with a specific electrochemical purpose. The extension is called a pilus, plurally pili, a term that covers a lot of microbial territory, because many bacteria grow pili for many different reasons: attachment, motility, genetic exchange. Geobacter's pili are different in a way that took researchers years to characterize properly, and the characterization is still not entirely settled, which is worth knowing up front. This chapter is built on real science. Some of that science is still being argued about in print. What seems solid? These filaments conduct electrons. Try for a moment to imagine a hair from your own head. Not to look at, to hold. The way it feels between 2 fingers, the barely there resistance of it, the fact that it has a thickness you can just barely register as texture rather than nothing. Now divide that thickness by somewhere between 500 and 1,000. Keep dividing until what you're holding has no business being called a physical object anymore. Until the concept of thin stops meaning anything useful. That's approximately the diameter of a Geobacter pilus. 3 to 5 nanometers. A nanometer is 1/1000th of a meter. The pilus is, in the most literal sense, not something you could interact with through any normal channel of human sensation. You could not feel it. You could not see it without electron microscopy. You could not confirm its existence through any means available to unaided human perception. And it conducts electricity. The conductivity, this is where the argument in the literature lives, and it's worth spending a moment here rather than skipping to the cleaner version. Early work, including influential studies from Lovely's own lab, characterized the pili as metallic-like conductors, suggesting electron transport happening along the length of the filament through a mechanism resembling what happens in conductive polymers. Later work complicated this. Some researchers proposed that the conductivity was mediated differently, through cytochrome proteins associated with the filament, rather than through the filament structure itself. The debate produced a lot of careful experiments and a few sharp exchanges in journal commentary sections. What hasn't been seriously disputed? When Geobacter grows these filaments in contact with an electron acceptor, iron mineral, electrode, another cell, current flows. Something is conducting. The filament is part of why. For the purposes of tonight, that's enough. What the pilus allows is distance. A Geobacter cell sitting in sediment is surrounded by mineral surfaces, organic compounds, other cells, pore water. The ferric ion it needs to reduce isn't always immediately adjacent. The cell can't move toward it the way a larger organism might reposition itself. What it can do, what the pilus makes possible, is extend its respiratory reach into the space around it, making contact with iron minerals several cell lengths away, conducting electrons along the filament to where the terminal reductase can complete the transfer. The cell stays, the electron travels. There's something in that arrangement that feels to me like a solution arrived at through a very long process of elimination. Not clever, not designed, just what remained after everything that didn't work stopped being tried. billions of years of anaerobic organisms encountering the problem of an electron acceptor that isn't close enough, and what persisted in this lineage was the filament, the reach. Your neurons do something structurally analogous, and this comparison is imprecise enough that it needs immediate qualification. Axons extend from neuronal cell bodies to make contact with targets the cell body itself doesn't reach: a muscle fiber, another neuron, a gland. The electrical signal travels along the axon. The cell body stays where it is. The reach is the function. Geobacter's pili are not axons. The mechanisms are different, the scale is different. The evolutionary history is entirely separate. But the geometric logic, extend, bridge distance, deliver charge, appears in both. Which probably says something about the limited number of solutions available when a cell needs to move electrons somewhere it cannot physically go, or maybe it says nothing general at all. Maybe it's a coincidence of description rather than a real parallel. I genuinely don't know. I find I think about it anyway. In a biofilm, a community of Geobacter cells growing together on a surface, the pili creates something the literature sometimes calls a conductive matrix. Individual cells connected through overlapping filaments, passing electrons not just from one cell to an iron mineral, but potentially from cell to cell, distributing the respiratory load across a community of organisms in contact with a shared electron acceptor. The biofilm conducts, not like a wire, not efficiently in any engineered sense, but measurably, verifiably. When researchers grow Geobacter biofilms on electrodes and measure the current produced by cells at different distances from the electrode surface, they find that cells far from the surface, cells with no direct contact with the electron acceptor, are still contributing to current production. Something is carrying their electrons to the surface. The filaments, the matrix, the reach, extended not just from one cell outward, but through a community of cells, each one connected to the next, the electrons finding their way to the surface through whatever conductive path the biofilm has assembled. It is fully dark where this is happening. That's not a poetic observation, it's a physical fact worth holding. The sediment layer where Geobacter builds its filaments and reduces its iron receives no light. Photons are not part of the energy budget here. The cell has no relationship to light, no photoreceptors, no behavioral response to the presence or absence of it. It grows its filaments in complete darkness, extends them nanometers into the surrounding mineral environment, conducts electrons along structures thinner than any human technology was producing until very recently, and does all of this through chemistry alone, through the same basic logic of electron gradients and membrane proteins that your mitochondria use right now, as you breathe, as your chest rises and falls with a slowness that sleep is already beginning to shape. The filament extends, the electron moves, the iron accepts it, and somewhere in the sediment beneath water you have almost certainly stood near this transaction is completing itself, quietly in the dark, at a scale so far below the threshold of human perception that the only honest response is a kind of stillness, not awe exactly, something quieter than that. Before bioluminescence, before the electric eel, before any organism evolved a nervous system capable of producing the coordinated electrical signals we call action potentials, before any of that, electrons were moving through living cells in ways that were, by any reasonable definition, electrical. Not metaphorically electrical, not analogously electrical. The electron transport chains running in the membranes of ancient anaerobic bacteria were moving charge across concentration gradients, generating electrochemical potential, doing work through the controlled flow of electrons from one molecule to another. The physics of that process is the same physics that runs through a copper wire. The medium is different. The molecular machinery is biological rather than metallic. But the underlying phenomenon, charge moving through a material in response to a gradient, is not different in any fundamental way. Geobacter didn't invent this. Nothing invented this. It emerged, in the way that chemistry tends to find the paths available to it, long before the word electricity existed in any language. There's a specific quality to old things that I find hard to articulate without reaching for something imprecise. Not reverence exactly, more like the feeling of holding an object that has passed through too many hands to track, a worn wooden handle, a stone tool, something whose age you can sense without being able to explain how. The electrons moving through Geobacter's membrane proteins tonight carry something like that quality, if you let yourself feel it rather than just know it. These proteins, the cytochromes, the iron-sulfur cluster proteins, the quinones shuttling electrons between complexes, are not new. Their basic architecture traces back to some of the earliest cellular life on Earth. The details have changed. The organisms carrying them have diversified beyond counting. But the core logic of the electron transport chain, the sequential passing of electrons down a thermodynamic gradient to a terminal acceptor, has been running with remarkable structural consistency for somewhere between 3 and 4 billion years. Geobacter's version extended this logic outward, past the cell membrane, into the mineral environment. That extension is maybe 2 billion years old, give or take. Here's what bioelectricity actually is, at the level where it stops being a concept and starts being a physical process. A membrane separates two environments with different ion concentrations. Proteins embedded in that membrane control what crosses it and when. Electrons, stripped from organic compounds through oxidation, enter the transport chain and are moved through a series of protein complexes, each transfer releasing a small amount of energy. That energy is used to pump protons across the membrane, building a concentration gradient. The protons flow back through a protein called ATP synthase, and that flow drives the synthesis of A to P, the cell's primary energy currency. The electron at the end of this needs somewhere to go. It needs a terminal acceptor. In your mitochondria, that acceptor is oxygen. 4 electrons, 4 protons, 1 oxygen molecule, water, clean, efficient. The reason aerobic respiration generates so much more ATP than the alternatives, in the sediment, in the dark, where oxygen ran out millimeters below the surface. Ferric iron. The interesting thing, and this took me a while to sit with properly, is that ferric iron is a worse acceptor than oxygen in strictly thermodynamic terms. The energy yield of iron reduction is lower. Geobacter's metabolism generates less ATP per molecule of organic carbon oxidized than your cells do. It works anyway. It has worked in anaerobic environments for long enough that the concept of working starts to feel inadequate. What would it even mean for a metabolic strategy to fail across 2 billion years of changing planetary chemistry, fluctuating mineral availability, mass extinctions, the rise and dominance of oxygen-producing photosynthesizers that fundamentally altered the atmospheric composition of the planet? Geobacter's lineage is still here. The strategy persisted. What bioluminescence does, the cold light produced by luciferase enzymes in fireflies, in deep-sea fish, in certain fungi, is make biology's relationship to energy visible. You can see it. A firefly's flash is electrons moving through a chemical reaction and releasing photons. People find it beautiful. It gets documented, studied, written about in ways that anaerobic iron reduction never quite attracts. And I understand why. Light is visible, beauty is a reasonable response to visible things, but the electron transfers happening in Geobacter's membrane proteins tonight. In the lightless sediment of a lake or a wetland or a waterlogged field, those are moving energy too. Capturing it, using it, releasing it in forms that cycle through sediment and water and atmosphere, and eventually back into the organic compounds that the next generation of cells will oxidize. No photons, nothing visible, just charge moving through protein in the dark, the way charges move through living membranes since before the Earth had free oxygen in its atmosphere. Your own bioelectricity is running right now, slower than when you're awake, but running. The neurons in your auditory cortex processing whatever you're listening to tonight. The ion channels in your cardiac muscle cells cycling through their depolarization and repolarization with the steady reliability that keeps the rhythm. The mitochondria in nearly every cell in your body passing electrons to oxygen, making water, making ATP, making it possible for you to breathe without thinking about breathing. Electricity, in the most literal sense. The same phenomenon in the sediment beneath a lake, extended slightly further, past the cell membrane, along a filament current to an iron mineral sitting in the dark. What separates these two versions of bioelectricity is mostly context. Organism, environment, electron acceptor, evolutionary history. The underlying physics is close enough that researchers studying microbial fuel cells use the same electrochemical vocabulary they'd use describing any other circuit, which is either remarkable or simply true depending on how you're holding it tonight. The first chapter of this narration began with sediment. Dark, cold, chemically patient. This one ends there too. Not because the circle needed closing, but because the biology keeps returning to that place. The substrate, the anaerobic layer, the place where oxygen runs out and something else takes over. Geobacter's bioelectricity isn't running toward anything. It isn't producing light or heat or any effect you could detect by standing near it. It's just completing its circuit. Electrons in, electrons out, energy captured in the middle, ion changed in the process, before the word existed for it. After the word stops mattering, still running. Uranium doesn't announce itself in groundwater. It has no color, no smell. It doesn't change the taste of water in any way a person could detect without instrumentation. It moves through an aquifer the way any dissolved ion moves, carried by the slow migration of groundwater through porous rock and sediment, spreading outward from its source, crossing boundaries that have no relevance to its chemistry. In Rifle, Colorado, the source was a former uranium ore processing mill. The mill operated through the mid-20th century and left behind, in the soil and groundwater beneath and around it, a contamination plume that didn't stop at the fence line. Dissolved uranium in its oxidized soluble form U6+, had entered the aquifer. It was moving. The problem with dissolved uranium, beyond the toxicity, is the moving. A contaminant that stays in one place can, in principle, be contained, excavated, managed at its source. A contaminant that migrates through groundwater requires a different kind of thinking, because groundwater doesn't ask permission to cross property lines, doesn't respect remediation boundaries, doesn't stop migrating because a cleanup effort has begun somewhere upstream. In 2007, researchers working at the Rifle site began an experiment, not a dramatic one, patient and procedural and easy to misread as unambitious. They began injecting acetate into the contaminated aquifer. Acetate, a 2-carbon organic compound, the same molecule Geobacter preferentially oxidizes in sediment. The logic was straightforward, at least in outline. The aquifer sediment already contained Geobacter and related iron-reducing bacteria, present in low numbers, metabolically slow in the absence of a rich carbon source. Acetate would feed them. A fed population would grow, a growing population would reduce more ferric iron, and critically would also reduce U6 plus to U4 plus. Oxidized uranium soluble mobile, reduced uranium insoluble immobile. It precipitates. It stops moving. The experiment ran. The uranium concentrations in monitoring wells downstream of the injection zone dropped. Not instantly, biology doesn't move at engineering speeds, but measurably over weeks, in a pattern consistent with microbial reduction rather than dilution or other geochemical processes. There's something worth pausing at here, which is how little was introduced. No organism was engineered. No novel chemistry was invented. No external biological agent was imported into a foreign environment. The acetate fed what was already present. The Geobacter, already in the aquifer sediment, already capable of uranium reduction, already running its electron transport chain at whatever rate the available carbon allowed, simply ran faster. The remediation, such as it was, came from accelerating a biological process that the aquifer was already, very slowly, doing on its own. I find that fact more interesting than the result, if I'm being direct about it. Not the cleanup, but the implication underneath it, that the chemistry needed for uranium immobilization was already present in the contaminated system. Had been present, presumably, since before the contamination arrived. The organism and the metabolic capability were there, they just needed more carbon. The experiment also revealed complications, as field experiments tend to. Acetate injection stimulated sulfate-reducing bacteria alongside Geobacter, which produced hydrogen sulfide and created secondary geochemical issues. When acetate injection stopped, uranium concentrations began to rebound, because the Geobacter population declined without the carbon subsidy, and the oxidizing conditions of the aquifer slowly reconverted immobilized uranium back to its soluble form. The immobilization wasn't permanent. The biology required maintenance, which is, honestly, how most biology works. Steady-state systems maintained by continuous process, not fixed outcomes. The aquifer's chemistry wasn't cured, it was for a period redirected. Rifle, Colorado. A former mill site, an aquifer with a contamination plume, a research team injecting acetate through wells into saturated sediment, measuring uranium concentrations in monitoring points downstream, watching the numbers move. Not dramatic, not fast, not a story with a clean ending, but real, verifiable. The data exists in published literature. The site coordinates are a matter of public record, and somewhere beneath the surface of that particular stretch of Colorado ground, Geobacter is still present in the sediment, metabolizing at whatever rate the available carbon allows, reducing whatever iron and uranium its electron transport chain encounters, indifferent to the history of the site above it. The contamination came from human industry. The partial remedy came from a bacterium that has been reducing metals in anaerobic sediment since long before the concept of industry existed. That asymmetry doesn't resolve into anything tidy. It just sits there in the sediment, the way facts tend to sit when you stop trying to arrange them into a lesson. The uranium that stopped moving because an electron transferred, one cell's respiration, multiplied across a population, across weeks, across a monitoring network designed to detect exactly this kind of slow, quiet chemical change. The groundwater kept moving. The uranium for a time did not. Nothing. That's the honest answer. From inside the cell, contamination looks like nothing, because the cell has no capacity for looking, no sensory apparatus tuned to the concept of contamination, no internal state that registers the difference between a uranium ion and any other reducible metal encountered in the course of running its electron transport chain. A Geobacter cell encountering U6+ in an aquifer sediment is not encountering a problem. It is not recognizing a foreign substance. It is not doing anything that could reasonably be called remediation from its own perspective, because it has no perspective in any meaningful sense of that word. What it has is chemistry, membrane proteins, a redox potential, a terminal reductase capable of accepting electrons from more than one kind of metal ion. The uranium fits the chemistry. The reaction runs. Sit with that for a moment, if you can, before moving on. There is a tendency, it's almost automatic, I think, and I notice it in myself, to import intentionality into biological processes that are working at scales and through mechanisms that have nothing to do with intention. A cell reduces uranium and an aquifer becomes less contaminated, and the human mind wants to draw a line between those two facts that passes through something like purpose, or like the cell was doing something for a reason. It wasn't. The electron transferred because the thermodynamics allowed it. The uranium precipitated because its oxidation state changed. The contamination decreased because enough cells ran enough reactions over enough time to shift the local geochemistry. The cause and the consequence are real. The purposefulness between them is something we added. If you could shrink, not realistically, just as a thought, the kind of thinking that happens easily when you're tired and the boundary between imagination and understanding gets softer. If you could shrink to the scale of a Geobacter cell and find yourself inside it, what you'd encounter first is density. Not darkness exactly, because darkness implies the absence of something that was expected. Light was never expected here. What you'd find is a space packed with protein, with lipid membrane, with ribosomes smaller than the wavelength of visible light, with DNA folded into a nucleoid region without the histone scaffolding your own chromosomes use. The cytoplasm is not empty space with organelles floating in it. It's more like a crowded, aqueous gel, every cubic nanometer occupied by something functional. The electron transport chain proteins are embedded in the inner membrane. You'd be very close to them. Close enough that the concept of close stops working properly, because at this scale, proximity and contact start to mean the same thing. An electron enters the chain. It moves. Not like a ball rolling, more like a probability redistributing itself across a series of protein complexes, each transfer releasing a small packet of energy that the cell uses to pump a proton across the membrane. The proton gradient builds. A TP synthase turns. A TP is made. At the end of the chain, an electron reaches the outer membrane cytochromes. Beyond them, through a pilus or directly through the mineral contacting proteins, a uranium ion, U6+. 2 electrons arrive. The uranium becomes U4+, insoluble. It leaves solution. The cell didn't know it was uranium. What the cell does respond to, and this distinction matters, is the availability of electron donors and acceptors. Acetate concentration in the surrounding pore water affects gene expression. When carbon is abundant, Geobacter upregulates the machinery for processing it. When electron acceptors are scarce, other regulatory responses occur. These are genuine biological responses to environmental conditions. They are not awareness. They are chemistry responding to chemistry. Protein concentration shifting in response to molecular signals, feedback loops operating without anything that could be called a decision. The distinction between response and awareness is one I find genuinely important, not just philosophically but biologically, because collapsing that distinction leads to a kind of storytelling about microbes that sounds compelling and is quietly wrong. The cell is not clever. It is not strategic. It is not in any sense trying. It is running. That's different. And it's enough. The mismatch in scale between what the cell is doing and what results from it. This is the part that keeps surfacing. The more time I spend with this organism, One Geobacter cell reduces some uranium ions in its immediate vicinity. The effect is local, chemical, small beyond any useful visualization. Now multiply that cell by 10 million, by a billion. By the population density of an active Geobacter community in acetate-amended aquifer sediment, running its reductions continuously across weeks of field experiment. The uranium plume slows. Monitoring wells show concentration decreases. A contamination event that entered the aquifer through industrial activity, that spread through groundwater without regard for property lines or remediation timelines, it is being altered incrementally by the aggregate of individual microbial respiratory events, none of which had any relationship to the plume as a concept. The cell knew about the acetate in its immediate pore water. It knew about nothing else. There's a quality to processes like this that I don't have a clean word for. Not beauty. That's too easy and slightly wrong. Not efficiency. The process is slow and metabolically costly, and produces secondary complications that require their own management. Something closer to the feeling of finding out that something you assumed was inert has been quietly doing something consequential the whole time. The aquifer sediment looked like sediment. It was a microbial community with a metabolic capability that happened to intersect with a human-made problem in a way that a human-made solution could leverage. No engineering of the organism, no modification of the chemistry, just acetate, injected into wells, feeding a population that was already there, already running its electron transport chain, already capable of the reduction that would slow the plume. The cell didn't know it was being used. It didn't know anything. It ran its chemistry in the dark, in the cold, in the contaminated groundwater of a former mill site in western Colorado, and the electrons moved, and the uranium stopped moving, and the two facts are connected by nothing more intentional than the thermodynamics of reduction, which is, depending on how you're holding it tonight, either deflating or quietly extraordinary. Maybe both. The cell is still running. Autumn, somewhere in the northern hemisphere. A lake. The surface temperature dropping by a degree or two over the course of the week. The kind of change that's too slow to feel if you're swimming, but shows up clearly in a temperature logger anchored to a buoy. Algae that bloomed through summer are dying back. Their cells, full of carbon fixed from atmospheric CO2 through photosynthesis, full of lipids and proteins and nucleic acids assembled over a season's worth of sunlight, are sinking, slowly. Aggregate particles of dead organic matter drifting downward through the water column, through the thermocline, into the cold and increasingly oxygen-depleted water near the bottom. They land in sediment. And then Geobacter, among others, begins the long work of taking them apart. Carbon doesn't disappear when it enters sediment. This is worth saying plainly because the intuition that things which sink and settle are somehow gone, removed from the system, finished, is wrong in a way that matters for understanding what anaerobic decomposers actually do. What sinks into a noxic sediment enters a processing queue. The organic carbon in those algal cells, in the fatty acids, the amino acids, the sugars, is still carbon, still chemically available, still connected to the planetary carbon cycle by the slow, continuous work of microbial metabolism. Geobacter's role in this queue is specific. It handles the acetate, the 2-carbon organic acid that other fermenters produce when they begin breaking down more complex molecules. The fermenters work first, cleaving larger compounds into smaller ones, and the products of that fermentation, including acetate, become Geobacter's substrate. Geobacter oxidizes the acetate, strips its electrons, passes them to iron. The carbon, now fully oxidized, leaves as CO2. It dissolves in the pore water, moves upward through the sediment column, eventually reaches the water column, eventually reaches the atmosphere. eventually possibly gets fixed again by a photosynthesizing cell somewhere on the surface. The loop is slow, months, years, decades depending on burial depth and sediment conditions, but it closes. You exhaled just now, or a few seconds ago, or you're about to, in the slow respiratory rhythm that sleep is already beginning to shape. The CO2 in that breath came from glucose your cells oxidized, glucose that came from food that came from plants or animals that came from plants that fix carbon from the atmosphere through photosynthesis. Some fraction of the carbon atoms in your body have passed through sediment before, have been buried, processed by anaerobic bacteria, released as CO2, fixed again by a photosynthesizer, eaten, digested, incorporated. The cycle is old enough and large enough that the odds of your carbon being genuinely virgin, never having passed through a microbial decomposer, never having sat in anoxic sediment, are essentially zero. Geobacter, or something very much like it, has handled your carbon before, probably more than once. What Geobacter contributes to this cycle is not unique. Other anaerobic bacteria perform dissimilatory ion reduction. Other organisms decompose organic matter in anoxic environments, but it is substantial. In iron-rich freshwater sediments, which cover a significant fraction of the Earth's wetland and lake bottom area, Geobacter and its relatives are primary decomposers. They handle a meaningful percentage of the organic carbon that enters those sediments. The numbers, when you look them up, are large in the way that biogeochemical numbers tend to be. Teragrams of carbon per year, global flux estimates with error bars wider than some countries. I won't recite them here. They don't land at this hour the way they land in a paper. What matters is the order of magnitude, significant, not trace, not marginal. A real and measurable contribution to the global carbon budget, coming from organisms that most people have never heard of, in sediments that most people have never thought about. There's a particular kind of Tuesday afternoon feeling to all of this that I find oddly comforting. Not the drama of it. There is no drama. Just the routine. Carbon falls, carbon gets processed, carbon leaves, carbon returns. The organisms doing the processing don't vary their approach based on the season or the weather or what's happening in the water column above. They run their metabolism at the rate that temperature and substrate availability allow. Winters the flux slows, some springs it accelerates. The sediment doesn't mark these fluctuations as significant. The chemistry adjusts and continues. Below the lake where the autumn algae settled, below the sediment layer where Geobacter is running its oxidations tonight, are older layers. Last year's organic matter, partially processed. The year before that, further along. Decades of accumulated material in various stages of decomposition, each layer slightly more altered than the one above it. The carbon in the deepest layers having spent years in the dark being slowly disassembled by communities of anaerobic bacteria working at whatever pace the chemistry allows. Some of that carbon will never fully leave. Some fraction gets buried deeply enough under enough pressure over enough time that it eventually becomes something else. Kerogen, eventually petroleum, eventually the kind of carbon that stays locked in rock for millions of years until something extracts it. But most of it cycles. Most of it eventually becomes CO2 and returns. The autumn algae that sank 3 weeks ago in a lake somewhere in Michigan or Ontario or Scandinavia, some of their carbon is already moving through Geobacter's electron transport chain tonight, being stripped of electrons, being passed to iron, being released, going somewhere, coming back eventually as something else. The lake surface, meanwhile, is cold and still. Whatever is happening in the sediment below it is happening in a place that has no surface expression, no visible sign, nothing a person standing on the shore would be able to detect. The decomposition is invisible. The carbon is moving. Your breath slow now is part of the same system, just a different part, running at a different speed through a different set of biochemical reactions toward the same eventual outcome. Carbon doesn't disappear, it just goes somewhere you're not watching. Two cells, different species, no nervous systems, no signaling molecules passing between them in any conventional communicative sense. No shared evolutionary history that would predict this particular chemical arrangement. And yet electrons are moving from one to the other, not randomly, through a specific sustained thermodynamically favourable pathway that the two organisms maintain together in anaerobic sediment at a metabolic rate that neither could sustain as efficiently alone. The arrangement persists because it works, not because anything chose it, because the cells whose metabolism produced it left more descendants than the cells whose metabolism didn't. That's the whole story at the base of it. Everything else is detail. The formal term is direct interspecies electron transfer, or DIET. It was characterized relatively recently, the key papers establishing it as a genuine phenomenon rather than an artifact of experimental conditions appeared in the 2010s, and the research is still expanding. Geobacter is central to it, both as an electron donor in some configurations and as an electron acceptor in others, depending on what's available in the community and what the thermodynamics of the local environment favour. In methanogenic communities, the anaerobic microbial consortia that produce methane as a metabolic byproduct, Geobacter has been observed forming direct electrical connections with methanogenic archaea. The Geobacter oxidizes organic compounds, generates electrons, and rather than passing those electrons to iron, passes them directly to the archaeon. The archaeon uses them to reduce CO2 to methane. Two organisms, one electron pathway. The methane leaves the sediment, enters the water column, eventually reaches the atmosphere. For a long time, the understood mechanism for interspecies electron transfer was chemical. Organisms exchanging hydrogen gas or formate as electron carriers, molecules that diffuse through solution from producer to consumer. This is called INDE, direct interspecies electron transfer, and it's real and well documented and still happens extensively in anaerobic communities. But hydrogen diffusion is slow. It's limited by concentration gradients. In dense microbial communities where metabolic rates are high, hydrogen transfer can become a bottleneck. The producing organism has to wait for the consuming organism to clear the hydrogen before it can produce more, and the whole consortium slows. Direct electron transfer bypasses this. The electron moves through a conductive connection, through Geobacter's pili, or through conductive mineral particles like magnetite that the organisms use as intermediaries, without becoming a diffusible molecule at all. No hydrogen produced, no hydrogen waiting to diffuse, just charge, moving from one cell's metabolism directly into another's. The consortium runs faster. I keep wanting to call this a partnership and then stopping myself because the word carries implications that the biology doesn't support. Partners, in the way the word usually functions, have some form of mutual recognition, some capacity to enter and maintain a relationship. What Geobacter and a methanogenic archaeon have is a thermodynamic arrangement that persists because abandoning it would cost both organisms metabolic efficiency. It's sustained chemical dependency without anything that resembles awareness of the dependency. Which is, I'm not sure if this makes it more or less interesting. I've turned it over enough times that I've lost my original intuition about it. What I keep coming back to is that the dependency is real regardless of whether the organisms experience it. The consortium's function depends on the connection. Remove Geobacter from the community, and the methanogen's metabolic rate changes. The relationship has consequences even if it has no interiority. That might be enough to call it a relationship. With the right qualifications, magnetite complicates this further, and in a way I find genuinely strange. Magnetite, Fe3O4, is a conductive iron mineral that Geobacter produces as a byproduct of its iron reduction activity. Small magnetite particles accumulate in sediment where Geobacter is active, and those particles, it turns out, can serve as electron conduits between organisms that aren't in direct physical contact. A Geobacter cell and a methanogenic archaeon, separated by a small distance both in contact with a magnetite particle, electrons can move from the bacterium through the mineral to the archaeon. No pilus required, no direct cell-to-cell contact. The mineral is doing the conducting. The mineral that Geobacter produced through its own metabolism. What this means for how we think about microbial communities in iron-rich anaerobic sediments is that the conductive matrix isn't purely biological, it's partly mineral. The metabolic activity of Geobacter creates the physical conditions, the magnetite particles, the conductive biofilm components, that enable more efficient electron transfer within the community it's part of. The organism is, in a sense, building its own infrastructure, except that framing implies intention, and we've already established that intention isn't part of this. What's more accurate, Geobacter's metabolism produces magnetite as a consequence of iron reduction, and magnetite's conductivity happens to facilitate electron transfer between organisms, and the communities where this occurs are more metabolically efficient than communities where it doesn't occur, and so those communities persist and those organisms remain associated. Consequence without plan. Infrastructure without an engineer. In the sediment of a methane-producing wetland tonight, somewhere boggy and waterlogged and smelling faintly of the sulphurous edge that anaerobic chemistry tends to produce, this is running. Geobacter cells passing electrons to archaea, methane forming and bubbling slowly upward through sediment layers, magnetite particles sitting at the junction between two organisms that will never know they're connected. The methane that reaches the surface was made in part by a connection that looks almost like a circuit. Two cells, an electron, a conductive path between them. No word for what they are to each other that doesn't import something false, just the electrons moving and the methane rising, and the sediment doing what sediment does when the right organisms are present and the carbon is available and the thermodynamics allow, which is in the end all it's ever needed to do. An electrode is, at its simplest, a surface that conducts electrons. Stainless steel, carbon cloth, graphite. Something with enough conductivity to accept charge and move it somewhere useful, into a circuit, toward a measurement, through a wire thin enough to run across the bottom of a river without disturbing the sediment above it. When Geobacter encounters an electrode in anaerobic sediment, it doesn't encounter a human invention. It encounters a surface, conductive, oxidized enough to accept electrons, chemically available in the same basic way that ferric iron minerals are chemically available, something the terminal reductase enzymes at the cell's outer membrane can work with. So it works with it. The cell attaches, it begins oxidizing organic compounds. It passes electrons through its transport chain to the electrode surface. If the circuit is complete, if there's a wire running from that electrode to another electrode in an oxygen-rich environment, current flows, measurable current. Microamps typically, sometimes milliamps in optimized systems with dense biofilms. Real electricity from a bacterium that has never heard of electricity, growing on a surface it has no reason to distinguish from iron. The first microbial fuel cell demonstrations, systems deliberately designed to capture current from microbial metabolism, date to the early 20th century. But those early systems used mediator chemicals to shuttle electrons from the cell to the electrode, because researchers at the time didn't know that certain bacteria could transfer electrons directly. The mediator did the bridging work. When Geobacter's direct electron transfer capability was characterized, it changed what a microbial fuel cell could be. No mediator needed. Grow Geobacter on a carbon electrode in anoxic sediment, connect it to a cathode in oxygenated water above, and the organism's own respiratory process generates the current. No moving parts, no fuel input beyond whatever organic matter exists in the sediment. No maintenance in any conventional sense. The organism maintains itself as long as the organic carbon holds. Outside, or maybe it's just the particular quality of late-night quiet, there's a sound like a heating system clicking somewhere distant, metal expanding in a pipe, nothing that needs attention. The chapter keeps going. Practical applications of sediment microbial fuel cells have been modest. Genuinely modest, not in the self-deprecating way that researchers sometimes undersell promising technology, but in the accurate sense that the power outputs are small and the engineering challenges of deploying electrodes in real aquatic environments are significant. What they've powered: remote environmental sensors. Data loggers placed in locations where battery replacement would be difficult or dangerous. Deep-sea sediment monitoring stations. River bottom sensors tracking water chemistry over extended periods. The Geobacter biofilm on the electrode generates enough current to run a low-power sensor and transmit occasional data packets. Not enough for anything requiring sustained high power. Enough for something that needs to sit quietly in the dark and occasionally report what it's measuring. There's something I find quietly funny about this in a way that doesn't really go anywhere. An organism that has been running its metabolic electricity in anoxic sediment since before the Mesozoic, since before dinosaurs, before flowering plants, before fish had fully diversified into their modern forms, is now being asked to keep a sensor battery charged. It's a significant underutilization of something very old. The Geobacter doesn't mind, it doesn't have opinions about its deployment. It just grows on the electrode and runs its chemistry, and the electrons go where the circuit sends them, which is no different, from the cell's perspective, from the electrons going into iron. A surface accepted them. That's sufficient. The biofilm that forms on a microbial fuel cell electrode is worth picturing slowly. It's not a thin smear. In active systems, the biofilm can grow to tens or hundreds of micrometers thick, many cell layers, a community rather than a monolayer. The cells closest to the electrode surface have direct contact, their outer membrane cytochromes touching the conductive material directly. Cells further from the surface rely on the conductive pili, the nanowires extending through the biofilm matrix, carrying electrons from their metabolism toward the electrode they can't directly reach. The whole structure conducts. Outer cells passing electrons inward, inner cells passing them to the electrode, the current flowing continuously as long as organic carbon is available in the surrounding water. Dense, dark, electrically active in a way that no instrument you could hold in your hand would let you feel. The sensor it's powering somewhere at the bottom of an estuary is measuring salinity, or dissolved oxygen, or sediment temperature. Something quiet and continuous that nobody will look at directly for weeks, just a data stream accumulating on a server, flagged only if something goes outside expected range. The Geobacter biofilm on the electrode below it is thicker now than it was last month. The current is stable. What stays with me at the end of this chapter isn't the technology, it's the image of a biofilm growing in the dark on a carbon electrode, doing exactly what it would do on an iron mineral at a pace that has no relationship to the human timeline of the project that placed it there. The researchers who deployed the sensor have moved on to other work. The Geobacter is still growing, still oxidizing acetate and still passing electrons to whatever conductive surface the circuit provided, still running the same respiratory process it was running long before the electrode existed, long before the concept of a sensor existed, long before anything in the sediment above it had changed in any way the cell would have detected. If detecting was something it did, which it isn't, it just runs. The Earth was 4 billion years old before oxygen became a significant presence in its atmosphere. Sit with the arithmetic of that briefly, not to understand it. Understanding isn't quite available at that scale, but just to feel the shape of it. 4 billion years. The planet forming, cooling, developing oceans, developing chemistry complex enough to produce self-replicating molecules, developing cells, developing metabolisms, developing the first photosynthesizers, all of it through timescales that make the entire history of animal life look like a rounding error, in an atmosphere where oxygen was scarce enough to be essentially absent. Anaerobic metabolism wasn't a workaround. It wasn't a backup system for organisms that hadn't yet evolved something better. It was the primary mode, the default, the only mode for most of life's history on this planet. Geobacter's biochemistry exists inside that history. What the early Earth's atmosphere held instead of oxygen was mostly nitrogen, CO2, methane, water vapor, with hydrogen and other gases in varying concentrations depending on volcanic activity, on what was outgassing from the interior, on the complex and poorly constrained chemistry of a young planet still settling into its own geochemical steady states. There was no ozone layer. Ultraviolet radiation reached the surface at intensities that would be lethal to most modern surface life. The oceans were chemically different, more iron-rich, more reducing, the dissolved iron sitting in a state that oxygen-rich seawater would immediately oxidize and precipitate. In that ocean, in those sediments, organisms were running electron transport chains, moving charge across membranes, capturing energy, reducing metals, producing methane, running the basic biochemical logic of life through whatever terminal acceptors the environment provided. Iron was abundant, it was available, it accepted electrons. The organisms that could use it persisted. The Great Oxidation Event, the term given to the period roughly 2.4 billion years ago, when atmospheric oxygen levels rose dramatically, largely as a result of oxygenic photosynthesis by cyanobacteria, was, from one angle, a catastrophe. For strictly anaerobic organisms, oxygen is toxic. It reacts with iron-sulfur clusters in proteins, generates reactive oxygen species, disrupts the carefully maintained reducing conditions that anaerobic metabolism requires. The rise of atmospheric oxygen was, for the organisms that had been running the planet's chemistry until that point, something between a slow disaster and an extinction pressure. Most anaerobic lineages retreated into sediments, into hydrothermal vents, into the oxygen-depleted layers of stratified water bodies, into every niche where the new atmospheric chemistry hadn't yet penetrated. Geobacter's ancestors went into the sediment and stayed there. There is something I keep returning to in this, not grief exactly, nothing that's settled or resolved into a named feeling, more like the sensation of standing at the edge of something and not quite being able to see the bottom. The world that produced Geobacter's biochemistry doesn't exist anymore, not at the surface. The anoxic ocean, the iron-rich water, the atmosphere with its particular reducing character, these are gone in any form that a person could visit or touch or breathe. What remains of that world is chemistry, the electron transport chain running, in Geobacter's membrane tonight, uses protein architectures that are, in their deepest structural features, very old. The cytochromes, the iron-sulfur proteins, the quinone carriers, These protein families predate the Great Oxidation Event. They were running their chemistry in organisms that existed before oxygen changed everything, and they are still running in modified forms in organisms that retreated to the places oxygen never fully claimed. The sediment preserved them, not deliberately, because the sediment is where oxygen runs out, and the organisms that require oxygen's absence found in that environment a place where the new atmospheric chemistry hadn't changed the fundamental conditions of their existence. 2.4 billion years is not a number that lands. I've read it dozens of times in different contexts, and I'm not sure it has ever fully registered as a duration I can feel rather than just state. What I can access is something smaller. The idea that a protein family running in a Geobacter cell tonight has structural relatives, not identical, substantially modified by billions of years of evolution, but recognizable to a structural biologist in organisms that have been extinct longer than complex life has existed. The biochemistry is old in a way that the organism's appearance doesn't advertise. A Geobacter cell looks, under an electron microscope, like a small rod-shaped bacterium, unremarkable. The kind of organism you'd pass over in a sample if you weren't looking specifically for it. Nothing about its morphology announces that it's running chemistry with roots in the Archean. It doesn't announce anything. It just runs. What the sediment does, among its other functions, is maintain the conditions that make this possible. Below the oxic surface layer, the reducing environment that Geobacter requires is preserved. Not because the sediment is protecting anything, not because any process is maintaining it intentionally, but because oxygen consumption by aerobic organisms and chemical reactions in the upper layers depletes the oxygen before it can penetrate deeply. The anoxic zone in modern sediment is, in a limited but real sense, a remnant. A patch of early Earth chemistry persisting within a planet that has largely moved on to oxidizing conditions. Geobacter lives in that remnant, runs its ancient biochemistry in a microenvironment that resembles chemically the world its lineage evolved in. The retreat into sediment, 2+ billion years ago, turned out to be survivable. More than survivable. The sediment is, from the organism's perspective, if perspective applies, everything it needs, anoxic, iron-rich, organic carbon available. The conditions that made life possible before oxygen existed still exist. In every lake bottom, every wetland, every waterlogged soil on the planet. The Great Oxidation Event changed the surface. It changed the oceans slowly. It changed what was possible in the open atmosphere. It didn't fully reach the sediment. Geobacter is still there, in the part of the planet the oxidation didn't claim, running the chemistry that worked before the chemistry changed, passing electrons to iron in the dark at the pace that biology moves when nothing outside is asking it to be anything other than what it already is, what it has always been, what it will, in all likelihood, continue to be long after everything currently living at the surface has been replaced by something the sediment hasn't produced yet. Nothing. Again, nothing. And yet the word keeps feeling insufficient when you look closely at what a biofilm actually does. A single Geobacter cell attached to a surface behaves differently than a Geobacter cell floating free in solution. Its gene expression changes. Specific proteins are upregulated, proteins involved in surface attachment, in extracellular matrix production, in the biosynthesis of the conductive pili. The cell is not the same organism it was before contact. The surface changed something, through chemical signals too small and fast to follow, and the cell responded by becoming, in a real molecular sense, a different version of itself. Now multiply that by 10 million. The biofilm that forms is layered in ways that took researchers years to characterize properly. partly because the tools for looking inside a living biofilm without destroying it are difficult to develop and limited in resolution even now. What's known? The cells nearest the attachment surface experience different conditions than the cells at the biofilm's outer edge. Substrate concentration varies with depth. Organic carbon is more available at the outer surface, closer to the bulk fluid, and depletes inward. Metabolic activity varies accordingly. The cells deeper in the biofilm are running their chemistry under different constraints than the cells at the periphery, and their gene expression reflects this. Different zones, different metabolic states. One structure, many internal environments. It's not a uniform mass. It never was. The conductivity of the biofilm, this is the part that keeps snagging my attention, the part I find hardest to hold at the right distance. A single Geobacter cell conducting electrons to an electrode is one thing. A biofilm 60 micrometers thick, with cells at the outer surface passing electrons inward through overlapping pili networks toward the electrode they can't directly reach. That's something the word conductive doesn't quite capture. The current density produced by thick Geobacter biofilms on electrodes exceeds what you'd predict from the number of cells in direct contact with the surface. The cells that aren't touching the electrode are still contributing. Something is moving their electrons toward it. The pili, the matrix, the structure the biofilm assembled from its own metabolic activity, through no process that resembles planning, conducting charge across distances that individual cells cannot bridge alone. It starts to resemble, and this is where I have to be careful, because the comparison almost works and then doesn't. It starts to resemble a tissue. A layer of cells with specialized zones, coordinated function, internal gradients that shape behavior across the structure. Something like the way cells in a liver lobule experience different oxygen concentrations depending on their position relative to the blood supply, and adjust their metabolism accordingly. But a liver is the product of developmental biology. Coordinated gene expression guided by morphogen gradients, cell-to-cell signaling, the elaborate machinery of multicellular development that took hundreds of millions of years to evolve. A Geobacter biofilm assembled itself through attachment and growth and the local chemistry of the surface it's on. No developmental program, no body plan. The similarity in outcome, zonation, metabolic specialization, coordinated electrical activity, arrive through entirely different routes, which means the comparison is useful up to a point, and then it isn't. And the point where it stops being useful is roughly where it would start to feel satisfying, which is maybe the most honest thing I can say about it. Matrix. That's the word the literature uses for what surrounds the cells in a biofilm. The extracellular polymeric substances, the EPS, the mixture of polysaccharides and proteins and nucleic acids, and lipids that Geobacter and other biofilm-forming bacteria secrete into the space between themselves. The matrix holds the structure together. It maintains the spatial relationships between cells. It provides channels through which nutrients move and waste products leave. It also contains conductive components, proteins embedded in the matrix that participate in electron transfer, cytochromes sitting outside the cells in the extracellular space, passing electrons between cells that aren't in direct contact. The electricity is partly inside the cells and partly outside them, in the matrix, in the space the biofilm created between itself. That detail took me a while to absorb properly. The conducting isn't only through the pili extending from cells, it's also through the material the cells secreted around themselves, into which they are now embedded, which now carries their electrons as part of a shared electrical infrastructure that no single cell built or maintains. The biofilm on an electrode in a microbial fuel cell produces a characteristic current signature over time. In the first hours after inoculation, almost nothing. Then a slow rise as cells attach and begin expressing the proteins needed for surface-associated life. Then an acceleration as the biofilm thickens and the conductive matrix develops. Then a plateau, a steady-state current reflecting the metabolic capacity of the mature community. If you graphed it, you'd see something that looks biological rather than mechanical, not a switch being flipped, a system finding its working level, stabilizing, maintaining. The graph is not beautiful in any aesthetic sense. It's a curve on a chart. But there's something in its shape, the slow rise, the acceleration, the plateau, that carries the feeling of a process that knows, in whatever non-cognitive sense that word can be stretched to, what it's doing. It doesn't know. The curve just looks that way. Right now, somewhere, a Geobacter biofilm is at some point on that curve. Maybe newly formed, cells just beginning to upregulate their surface attachment proteins, the matrix barely started. Maybe mature and thick, the conductive network fully developed, electrons moving through pili and matrix from outer cells to inner cells to electrode current steady. Maybe aging, carbon depleted in the local environment, cells at the biofilm's base dying and lysing, the structure beginning to detach in patches and drift downstream, cells dispersing into solution to find new surfaces, the whole cycle beginning again somewhere else. The biofilm doesn't persist indefinitely. Nothing does. It forms because the conditions allowed it. It maintains itself while those conditions hold. When they change, when carbon runs short, when the surface is disrupted, when something in the local chemistry shifts past a threshold the community can't compensate for, it releases. The cells disperse. And somewhere else, on a different surface, in conditions that suit them, they attach again. What a biofilm knows is nothing. What a biofilm does is considerably more than nothing. More than the sum of its individual cells would accomplish alone. More than the conductivity of any single cell to electrode connection. More than the metabolic output of a population without the matrix, without the spatial organization, without the zonation that emerges simply from cells growing in layers on a surface and experiencing each at its own depth, the particular chemistry of the place it inhabits. The collectivity is real, the awareness is not. Both of those things are true simultaneously, and the chapter doesn't know what to do with that except to set it down carefully, the way you'd set down something fragile that you're not entirely sure you were meant to be holding. For most of the 20th century, the assumption held without much examination. Below a certain depth, the subsurface was sterile, not hostile to life in any dramatic sense, not actively lethal, just empty, too hot in places, too pressurized in others, too far from sunlight and organic carbon and the conditions that life, as it was then understood, required. The surface of the earth was where biology happened. Everything beneath that surface past the shallow soil horizon where roots and soil fauna operated was geology. Rock, mineral, chemistry without biology. The assumption was reasonable given what had been looked for and where and how. It was wrong. The first serious evidence that the deep subsurface harboured living organisms came from unexpected places, from water samples drawn from deep mines, from cores drilled through basalt, from fluid extracted from rock formations hundreds of meters below the surface. The organisms found they were sparse by surface standards, metabolically slow, running their chemistry at rates so reduced that generation times, the time from one cell division to the next, were estimated not in hours or days but in centuries per generation. The number lands differently depending on how tired you are when you encounter it. Tonight, at this hour, it might just settle without fully registering, which is fine. It doesn't need to register completely to be true. Geobacter and its relatives appear in deep subsurface samples, in aquifer sediments tens of meters below the surface, in the saturated zones of rock formations that haven't been exposed to surface conditions in thousands of years. They're not the only organisms present in these environments. The deep biosphere, as it came to be called, contains a genuine community. Iron reducers, sulfate reducers, methanogens, fermenters, organisms representing lineages that diverged from surface relatives long enough ago that their relationship to known taxonomy is sometimes uncertain. What connects them is the absence of oxygen and the presence of chemical energy in some form. Reduced minerals, organic carbon trapped in rock pores, hydrogen produced by geochemical reactions between water and certain rock types. Life in the deep subsurface runs on whatever the geology provides. It's been running on that for a long time. The estimates for the total biomass contained in the deep biosphere have been revised upward several times since serious study of it began in the 1980s and 1990s. Current estimates place it at somewhere between 15 and 23% of total Earth biomass, arranged with large uncertainty bars because sampling the deep subsurface is expensive and technically difficult, and the estimates rest on extrapolations from a relatively small number of sites. But even the lower end of that range is a significant fraction of all life on Earth, living in a place that was assumed, for most of the history of biology as a formal discipline, to contain no life at all. The assumption wasn't irrational. Nobody was being careless. The deep subsurface simply wasn't where people looked, and what you don't look for, you don't find, and what you don't find, you eventually assume isn't there. There's a particular quality to scientific discoveries that involve finding something where absence was assumed. Not the excitement of a new phenomenon exactly, but something quieter. The adjustment of a boundary. The redrawing of a line that had been treated as fixed. The line between the living and the non-living parts of the planet moved when the deep biosphere evidence accumulated. Not dramatically, not all at once. Paper by paper, core sample by core sample, the picture changed. The living portion of the Earth extended downward into rock that had been classified, without much thought, as purely geological. Geobacter was part of what was found down there, running its iron reduction in aquifer sediments at depths and under pressures that the organism has no awareness of, at metabolic rates calibrated to the scarcity of the environment, dividing, if the generation time estimates are accurate, at intervals that make the concept of population growth feel almost meaningless. A cell dividing once per century is, in most practical senses, not growing, it is maintaining, replacing itself at a rate just sufficient to compensate for mortality, persisting in a state that hovers at the boundary between active metabolism and something that would be, at the cellular level, almost indistinguishable from suspension. Almost, not quite. The electron transport chain is still running, slowly, at a rate matched to the trickle of organic carbon available in pore spaces in the rock. The iron is still being reduced. The cell is still alive in every technical sense, maintaining its membrane potential, repairing damaged proteins, running its chemistry at whatever rate the available energy allows. It has been doing this, in some cases, in rock formations that were last exposed to surface conditions during periods of Earth history that predate any organism currently alive at the surface. What sits underneath the ground you're closest to tonight, underneath whatever building or landscape surrounds the place where you're lying, is rock at some depth, and in the water-saturated pores of that rock and in the sediment layers above it, organisms are metabolizing. Slowly, in conditions that surface biology would find inadequate, on energy budgets that make the most ascetic surface existence look extravagant. They are not waiting for conditions to improve, they are not adapted to surface conditions and surviving underground temporarily. This is their environment. The rock, the pressure, the dark, the trickle of chemical energy from mineral surfaces. This is what they are built for, if built is even a word that works here. What they have persisted in across timescales that no living thing at the surface will approach. The deep biosphere doesn't announce itself. No surface expression, no visible sign in the rock or soil above it, no phenomenon that would alert a person walking on the ground that below their feet, extending downward through sediment and into rock. A community of organisms is running its chemistry in conditions that look to surface biology, like the absence of everything necessary. Geobacter is down there, in some of those formations, reducing iron at whatever rate the energy allows. In the part of the planet that was, for most of the time, humans have been studying life simply not imagined to be alive. Somewhere in the middle of learning about an organism like Geobacter, there's a moment where the list of what it does starts to feel unreasonable. Iron reduction, carbon cycling, uranium immobilization, interspecies electron transfer, biofilm conductivity, deep subsurface persistence, each function real, each one documented across decades of careful research, each one representing a genuine contribution to the chemistry of environments that cover a significant fraction of the Earth's surface. It's a lot for something most people have never heard of. And yet the functions aren't remarkable in any biological sense. Not to the organism, which has no sense of its own remarkability, and not really to the planet, which has been running these cycles for long enough that Geobacter's contribution is simply part of the steady state. The infrastructure is quiet because it has always been there. Things that have always been there tend not to announce themselves. Take nutrient cycling first, because it underlies everything else. In anaerobic environments, wetlands, lake sediments, waterlogged soils, the anoxic zones of estuaries, the decomposition of organic matter requires organisms capable of running respiration without oxygen. Geobacter is among the primary decomposers in iron-rich versions of these environments. It handles the acetate, the simple organic compounds that fermenters produce when they begin dismantling more complex molecules. It oxidizes them completely to CO2, closes that part of the carbon loop, keeps the decomposition process moving at a rate that the environment's productivity depends on. Without functional anaerobic decomposition, organic matter accumulates. Carbon buries instead of cycling. The productivity of the system, the rate at which nutrients become available for new growth, slows. Wetlands that seem to persist effortlessly are persisting because their decomposition is working, which means their anaerobic microbial communities are working, which means organisms like Geobacter are running their chemistry in the sediment below the waterline. The wetland looks like plants and water and birds. It runs on microbiology. Mineral transformation is less visible still. As Geobacter reduces ferric iron to ferrous iron across decades and centuries of activity in a sediment layer, the mineral composition of that sediment changes. Iron oxyhydroxides dissolve. Secondary minerals, magnetite, siderite, vivianite, precipitate. The texture of the sediment shifts. Its porosity, its capacity to bind other compounds, its chemical reactivity, all of these change as the iron mineralogy changes. Sediment is not inert. It is being written continuously by the biology living in it. The geochemistry of a lake bottom reflects, in part, the metabolic history of its microbial community. The iron minerals present in a core sample taken from a freshwater sediment carry a record, partial, complex, not easily read, of the biological activity that occurred at each depth. Geobacter's activity is part of that record. It doesn't sign its work. The mineralogy just changes. Phosphorus moves through iron mineralogy in ways that connect Geobacter's activity to nutrient availability in ways that took a while to trace properly. Ferric iron minerals in sediment bind phosphate. They hold it, keep it from entering the water column, limit the phosphorus available for algal growth in the water above. When Geobacter reduces ferric to ferrous iron, those minerals dissolve, and the phosphate they were holding releases. It enters pore water. It moves upward. In phosphorus-limited lakes, which is most lakes, this release affects productivity at the surface. The algae growing in sunlit water above an anoxic sediment layer are, in part, dependent on the phosphorus mobilized by microbial iron reduction meters below them. Geobacter, reducing iron in the dark, is connected to what grows in the light. The connection is indirect, chemical, operating across distances and timescales that make it easy to miss. But it's there, in the geochemistry, in the nutrient budgets of lakes that have been studied carefully enough for the link to show up. It's late. Later than it was at the beginning of this, and the attention available at this hour is a different kind of attention than the kind that reads papers and follows citations. It's softer, more willing to rest on a single fact rather than pursue the next one, more inclined toward the texture of things than the mechanism behind them. That seems right for this chapter. The infrastructure doesn't require close attention to function. It functions whether anyone is paying attention or not. Contaminant immobilization sits alongside nutrient cycling and mineral transformation as a third dimension of what Geobacter provides to its environments, and Chapter 6 covered uranium specifically, so this doesn't need to stay long on that ground. What's worth adding here is the generality. Uranium was the well-studied case because it was the contamination emergency that drove the research funding. But the same reduction chemistry applies to other metals: technetium, chromium, vanadium. In each case, the oxidized, soluble form of the metal can be reduced by Geobacter to an insoluble form that precipitates and stops migrating. The organism wasn't shaped by evolution to manage industrial contamination. It was shaped to reduce metals in anaerobic sediment. The contamination arrived later. The chemistry was already there. What holds all of this together, the carbon cycling, the mineral transformation, the nutrient mobilization, the contaminant reduction, is not complexity in any design sense. It's the simple consequence of one metabolic process running continuously across a very large area for a very long time. Oxidize acetate, reduce iron, repeat. The planetary consequences of that repetition are real and significant and mostly invisible to anyone not specifically looking for them. The wetland functions, the lake maintains its nutrient balance. The sediment's mineral composition shifts slowly toward the equilibrium that Geobacter's activity creates. The contamination, where it encounters a population with sufficient carbon, slows. Nobody arranged this. No system coordinates it. Geobacter cells in a Michigan wetland and Geobacter cells in a Siberian lake are not connected to each other in any functional sense. They are simply running the same chemistry in conditions that allow it, producing similar consequences at local scales that, summed across the planet's anaerobic environments, add up to something that planetary biogeochemistry depends on. The infrastructure is quiet because it doesn't know its infrastructure. The organism has no relationship to the functions it provides, no awareness of the carbon cycle it participates in, no connection to the phosphorus that reaches the surface water above it, or the uranium that stops moving in the aquifer around it, or the mineral record it leaves in the sediment below it. It runs its chemistry. The planet, in whatever loose sense that word applies to a biogeochemical system, uses the output. Nothing about this arrangement requires recognition or acknowledgement, or even awareness that it's occurring. It was running before anything existed to notice it. It will run after the last organism capable of noticing it is gone. Right now, not as a figure of speech, not as a way of making the biology feel immediate when it might otherwise feel abstract. Right now, in the literal present tense of whatever moment you're in, The electron transfer is completing. Somewhere in the sediment beneath water near you, or far from you, or beneath ground so saturated it sits at the edge between soil and water, ferric iron is becoming ferrous iron. The oxidation state is dropping from 3 to 2. An electron arrived from a carbon compound that no longer exists in the form it had an hour ago. The iron accepted it. The cell captured the energy released in that transfer, used it to pump a proton, used that proton to make ATP, used that ATP to continue being alive. The transaction is done. Another one is already starting. There's no ceremony in it. That's the thing that keeps surfacing at the end of a long time spent with this organism. No threshold moments, no biological equivalent of punctuation, no point at which the Geobacter cell marks a completion before beginning again. The electron transport chain runs continuously or it doesn't run at all. There is no pause between one transfer and the next that would register as a pause at any scale available to human perception. It just runs. Your breathing has slowed since the beginning of this. Not because anything instructed it to. Bodies do this on their own when the hour is late and the voice in the ear is unhurried, and nothing in the immediate environment is asking for a response. The diaphragm drops a little further. The interval between breaths extends by fractions of a second that add up, over the course of a long narration, to a rhythm that sleep is already beginning to claim. Your mitochondria are running slower too. Not stopped. They don't stop, not while you're alive, but calibrated to the reduced demand of a body moving toward rest. Electrons still moving through your inner mitochondrial membrane. Oxygen still waiting at the end of the chain. ATP still being produced, at a rate just sufficient for the maintenance work that continues even in sleep. The Geobacter in the sediment is doing something structurally similar, not metaphorically. The logic of the electron transport, the membrane gradient, the terminal acceptor, these are the same in both cells. The acceptor is different. The organism is different. The scale is different by several orders of magnitude. The logic is the same. The nanowire extends. You don't need to picture it precisely. It's enough to know it's there, thinner than any human technology was producing until recently, reaching into the dark between the cell and the iron mineral, conducting electrons across a distance the cell cannot physically cross. The filament is out there right now, in the cold, in the lightless space between a cell membrane and a mineral surface, doing the thing it does. The Potomac River sediment where Derek Lovely collected his samples in 1987 still contains Geobacter. This is not a remarkable statement. The organism is present in virtually every anaerobic iron-rich sediment on the planet, but there's something in the specific continuity of it that sits quietly. The sediment that produced the first described Geobacter isolate is still producing Geobacter. The cells alive in that sediment tonight are not the cells that were cultured in 1987. The generations have turned over many times, but the lineage is unbroken. The chemistry that Lovely noticed was anomalous has been running without interruption since before he collected the sample, and it is running now in the same river, in the same dark below the waterline, with no awareness that anything was ever discovered about it. The discovery happened to the scientists. The Geobacter, as this narration noted at the beginning, just kept going. It's still going. What this series of chapters has tried to do, and whether it succeeded, is not something I'll know, because the people for whom it worked are likely asleep before it ends, is stay close to the biology without turning it into a lesson. Geobacter is not a lesson. It's an organism. It reduces iron. It extends P. It forms biofilms and persists in deep rock and transfers electrons between species and immobilizes uranium and contributes to the carbon cycle of a planet that has never needed to know it exists. It has been doing all of this longer than complex life has existed. It will, in all probability, continue doing it long after whatever currently qualifies as complex life has been replaced by something the sediment hasn't produced yet. The sediment is dark, the water above it is cold or warm depending on the season, depending on the latitude, depending on things that the geobacter in the sediment below has no mechanism to register. The electron moves, the ion changes, the cell continues. You're somewhere in the drift between awake and not. That particular quality of attention where the words are still arriving, but the meanings are softer, less insisted upon, more willing to pass through without snagging. That's where this narration was always trying to leave you. Not with understanding exactly, with something quieter than understanding. The sense that somewhere, in the dark below water you've stood near, or imagined or will someday cross, something is running its chemistry at a pace that has no relationship to urgency. Slowly, without interruption, without need for witness, still running. If something in these chapters found you at the right moment, if the biology settled into the background in a way that let your mind finally stop producing its own noise, then this channel did what it was made to do. The Sleepy Biologist exists because enough people felt that science, delivered slowly in the dark at the pace biology actually moves, could be its own kind of rest. If that's been true for you tonight, a subscription keeps this space alive. A like tells whatever algorithm governs these things that the content found its audience. And if you're a channel member, or if you've ever left a comment at 2 a.m. about an organism you'd never heard of before, thank you. Genuinely. This narration exists because you kept coming back. The Geobacter doesn't need to be thanked. It just runs. You, on the other hand, you can sleep now.